Hey, Assalamu alaikum and hey guys, this is Jem here. This is my fifth A level biology video, and we're gonna learn about the part two of nervous system in this chapter. Today we're gonna learn about the ultrastructure of striated muscles. Before that, of course, we gotta know the simple structure of the striated muscles. Remember that every single muscle has a lot of muscle fibers in it. It's like a specialized unit, like every plant, every leaves have their cells, plant cells in them. Just like that, every muscle has muscle fiber in them. So this is a really essential part of our muscles, which is the muscle fiber. What is the muscle fiber made up of? Well, the muscle fiber contains a lot of contractile proteins. Contractile proteins means compact proteins, proteins that are, arranged in compact. They're compactly arranged. They're in strands. They're in strips, and the movement. It's according to each other. Like if one moves, the other is also gonna move. So, yep. The each muscle fiber is surrounded by a self-surface membrane. Remember that just like cells have every single part of them, like a cell surface membrane, like an endoplasmic reticulum, like a cytoplasm. These muscle fibers also have these parts. The cell surface membrane in the muscle fiber is called the sarcolemma. The cell cytoplasm is called the sarcoplasm. The endoplasmic reticulum is called the sarcoplasmic reticulum inside the muscle fiber. Now we're gonna get into the structure of a muscle fiber. I remember I didn't tell you, but I'm gonna tell you now that a striated muscle is always. What's the difference between a striated muscle and a normal muscle? The difference is that a striated muscle is a muscle that is attached to a bone. It has to have a bone attached to it, right? So if it doesn't have a bone, then it's not called a striated muscle. So before let me show you guys here that even before that there was a diagram here. Yeah, this one. You guys can see that this muscle is attached to the bone, right? These leg muscles in the glutes, muscles in their biceps, all these muscles are attached to all these muscles are attached to the bones. So now we're gonna learn over here that the muscle fiber. This is a bone. This is a muscle. So there's a tendon here, and therefore it's a striated muscle. So inside these muscles, I told you guys there are a lot of muscle fibers present. Now these muscle fibers are these long strips. I told you before that they're always in strips and strands. So this is a long strand of a muscle fiber, and they consist of these nuclei. They consist of mitochondria. They consist of sarcoplasm. They consist of a sarcolemma, which is the membrane, and they consist of sarcoplasmic reticulums. And each of the striated muscle have many of these, which is really important. So, why is this important? It's important for the movement of these muscles. These muscles, they're special in their movement of the striated muscles. The bones, they assist in the movement of these muscles. That's why they're attached to them. So now this is a more detailed structure of what is present inside inside the fibers. Well, you guys can see here that these are the mitochondria. These ones. This is the sarcolemma, the membrane on top of it. This is the T tubule. This one. And these are the sarco. This is the SR, the sarcoplasmic reticulum. Remember that the SR is always this type of structure, so this is pretty obvious that it's the sarcolemma. I mean the sarcoplasmic reticulum. Now these are myofibrils. Now these myofibrils are really important structures. You guys can see these dark spots inside. The light spots, the things that are what are these myofibrils present inside? You guys can see that these are spots, right? But what are they surrounded by? This is the cytoplasm of the of the of the of the fiber, and the cytoplasm is called the sarcoplasm here. So remember that the myofibrils are surrounded by the sarcoplasm. This is really important. Remember. So the myofibrils. The myofibrils are located in the sarcoplasm. As I told you guys, each myofibril is made up of two types of protein filaments. Remember these two types of protein filaments. They're essential for this chapter. The thick filaments and the thin filaments. The thick filaments are called myosin, and the thin filaments are called actin. So remember, these two filaments are really, really important. So the myofibrils are cylindrical in shape. So the Z line is actually a disc. You guys can see here these two lines, Z line, M line. Z line is the red color line. It's not always red. It's just shown in this diagram. That the Z line is the red line, and the M line is the brown line. Then again the Z line. This is a whole pattern, continuous. So remember that this is a cylindrical structure, and therefore these lines, they're basically circles. So these circles, they separate the different parts of the cylinder, right? So this is one sarcolemma, this is another sarcolemma. So these are, I mean, sarcomere. So one sarcomere is separated by another sarcomere. And it's like the two cytoplasms are separated from each other. There's a membrane between them. This membrane covers the whole cylinder, right? There's a whole circle here. Imagine a circle being here. So therefore, the circle is called the Z line, which separates one sarcomere from another sarcomere, and then there's an M line. We're gonna learn what is present inside the M line and the Z line right here. So this is the same structure, it's just flattened here. You guys can see Z line, M line, then Z line, then M line. And one Z line to another Z line is called a sarcomere. Always remember, one Z line to another Z line sarcomere. Just like in waves, we learned that from one crest to another crest is called a wavelength. So here, remember that one Z line to another Z line is called a sarcomere. Now, this is there are three types of bands in our body. One is the I band, one is the A band, and one is the H band in our muscles. Sorry. So the H band, A band, and I band. Now, what's the difference between them? Remember that the I band consists of the Z line and the thick filament. Sorry, the thin filament. Okay, before that, sorry, I'm sorry, I'm interrupting this one. Before that, we need to know about the structure of the thin and the thick filaments. You guys can see here that there's a really obvious difference between them. The thin filaments, they are these like they're really thin compared to the thick ones, right? You guys can see the dark brown is the thick one, and these thin filaments here, they're the thin. Another big difference between them is that the thick filaments, you guys can see here, they have heads on them, right? And these heads are facing like away from the M line. Always remember that the thick filaments have myosin heads. They're called myosin heads because the thick filament is the myosin. So the heads on the myosin is called the myosin head. Remember that are the myosin heads. These myosin heads are attached to the thin filaments. You guys can see here, some parts of it are attached to the thin filament, and these are the bands. Let's get into it now. The Z line. What does the I band consist of? The I band consists of always a Z line and some part of the thin filament. Now this thin filament, it consists of the I band consists of the thin filament till the thin filament attaches to the thick filament. You see here that there's a part of it where it attaches to the thick filament, the heads that attach to the thick filament. So this part till that part, I mean till this part starting the attachment, it's called the I band. One Z line and a thin filament. It's called the I band. And over here also on the left side you can see thin filament and actin, thin filament and Z line. It's called, always remember, it's called an I band. Now let's talk about the H band. What does the H band consist of? It consists of two sides of it, thick filaments, two sides of thick filaments till it attaches to the thin filament. Again, this is not included here. So thick filaments, two sides, right? One here, one left, one right, and one M line. One M line, two sides. In the Z line, there was only one side of the thin filament and a Z line. And I mean, in the I band, in the H band, there's gonna be two sides, one left, one right, and an M line. Always remember this. In the I band, there's one side, one Z line, and the left side. I mean, if you're seeing the right side, then we're gonna say one left side and one Z line. In the I band, in the H band, there's always gonna be two sides, left, right, of the thick filament. till the thick filament attaches to the thin filament, and one M line. Now the biggest band here is the A band. What does the A band consist of? Remember we left out these parts in which the thin filaments are attaching to the myosin heads. Now these parts included with the H band is called the A band. So what is included? The parts where they attach and the H band, yeah. So one H band and one and the two sides that attach to the in which the myosin and the actin are attaching together. Now these two sides and one H band makes up the A band. And I told you guys that one I band to another I band, one Z line to another Z line is the sarcomere. Remember these things, okay? These are really important things. Now we're gonna learn about the contraction of muscles, about the sliding filament theory. This is a really important theory in A levels, sliding filament theory. Okay, so where does it all start from? It starts from basically neuromuscular junction. What is a neuromuscular junction? A neuromuscular junction is basically a neuro means neurons, muscular means muscles. So the muscle consists of fibers, of course. So a neuromuscular junction is a junction between a neuron and a muscle fiber. So remember what is a neuromuscular junction? A neuromuscular junction is a junction between a neuron and a muscle fiber. Now, what happens here is that an action potential arrives at a neuromuscular junction. Now, when an action potential arrives at a neuromuscular junction, it causes the calcium ions to be released into the sarcoplasmic, out of the sarcoplasmic reticulum. Remember that this is tropomyosin and troponin, right? What is a tropomyosin and troponin? Basically, tropomyosin is it's these thin filaments, this thin filament, and tropomyosin is the thin filament the thick filament that's present here. You guys can see, right? This filament, the red color, the line going here, and therefore the troponin is the circle here. So therefore, so the troponin is the circles, the dark circles you guys can see, and the tropomyosin is the thin filament. I mean, not the thin filament, the line here. Sorry, my bad. The tropomyosin is the line, the red color line, and the troponin is the circles there. Now, and all of this is happening in the thin filament, which is the actin filament. Remember that this happens in the actin filament. This is the thick filament here. So what was I saying? That the calcium ions, this is the thin filament, and outside this thin filament, there's a lot of sarcoplasmic reticulums, right? Now, when the action potential arrives at the neuromuscular junction, this causes the calcium ion gates to open in the sarcoplasmic reticulum. When these calcium ion gates open, they cause a lot of calcium ions to move out of the sarcoplasmic reticulum. When they move out of the sarcoplasmic reticulum, they come and attach to this thin filament. Uh, what does this achieve? Where do they attach on the thin filament? They attach in these troponin molecules. Remember that the calcium ions always attach to these troponin molecules. Now, when they attach to the troponin molecules, what do they achieve? This causes the binding sites on the thin filament to be exposed. Now, the thin filament has a lot of binding sites, and these binding sites are exposed. Are they exposed? We don't need to learn that, but just know that when the calcium ions they attach to the troponin, they cause the movement of the tropomyosin and the troponin. I mean, the whole chain, it moves in such a way that the binding sites are exposed. Now, when these binding sites are exposed, these myosin heads, they're gonna. Remember, I told you that the myosin heads they face away from the M line and they have myosin heads. So these myosin heads are therefore gonna go and attach to the binding sites. You guys can see here that in step one, what's happening that the myosin heads is therefore attaching to the binding sites here. When this happens, this causes the thin filament to move backwards. Now you guys can see the rowing action, right? What is happening here is that compare these two structures. It looks like the thick filament is moving forward, right? It's not moving forward. What is happening is the thin filament is moving backwards. You guys can see this arrow. the thin filament is pulled towards the center of the sarcomere. On the left side, just imagine that there's a sarcomere here, and this thin filament is moving towards it. So we'll just say that due to the myosin head, the movement of the myosin head, this causes the thin filament to move towards the sarcomere. So what is happening when the myosin head binds to it? The thin filament moves towards the sarcomere. So in a sense, we can say that the thin filament is contracting because it's moving towards sarcomere. One end is coming closer to another end, so therefore there's contracting, right? So the thin filament moves backwards towards the sarcomere, and the ATP number three, step number three, is that the ATP is going to bind to the myosin head. Now here, what is happening is that when ATP binds to the myosin head, it causes the myosin head to detach from the thin filament. I mean, we have achieved what we are going to do, right? The thin filament, it moves backwards. When it moves backward, what do we need to do? We need more thick filaments to move the thin filament backwards, right? I mean, it's held in one place after contracting. Once it's moved backwards, it's held there. After that, we need more myosin heads to come and contract it further, right? So to contract it further, more thick filament come in, and in order to, I mean, it's not like the thick filaments they spawn; they're already present and they're used again and again, right? So just know that if the myosin head is attached there, it's gonna get detached and then it's gonna go back again, and it's gonna attach to a thin filament from some other place, and then it's gonna bring it forward. Just imagine of this as like you're holding a rope and you're pulling a rope towards yourself, right? So you're gonna pull some part of it towards yourself. It's not gonna go back. You're gonna pull it towards yourself, and then you're gonna move your hand forward and pull more rope towards yourself, right? So just no, it's just the same thing. It's like your hand is the myosin head, and the myosin head is going back and forth again and again to pull more of the rope, and the rope is basically the thin filament. It keeps on moving towards you. I think you guys. I mean, it's a pretty good example. So I hope you guys like understood that one. So what does the ATP do here? It moves. Basically, it's like you need energy for your hand to move. For your hand to reposition itself and to pull more of the rope. So the ATP is doing the same thing here. ATP is doing hydrolysis, it's undergoing hydrolysis, and it's releasing energy. This release of energy causes the myosin head to reposition itself. And when it repositions itself, it attaches to another, therefore, the cycle continues. So it attaches to another thin filament, causing more of the thin filament to contract and moving it towards the sarcomere, uh, causing the whole thin filament to contract. So this was the process, and I hope you guys understood it. Let's go through it once more. Calcium ions, active. The action potential arrives at the neuromuscular junction, causes the calcium ions to be released. The calcium ion gate channels open from the sarcoplasmic reticulum. When this happens, the calcium ions they leave the sarcoplasmic reticulum and attach to the troponin molecules here. When they attach to the troponin molecules, the troponin and the tropomyosin, they move in such a way that they expose the binding sites of the thin filament. When they expose the binding sites of the thin filament, the myosin head of the thick filament comes and attaches to the binding sites of the thin filament. When this happens, when this attaches to the thin filament, the binding head, when this attaches to the thin filament, it causes a rowing action, and due to this rowing action, the thin filaments move backward. You pull the rope, so it moves towards yourself. So therefore, what happens is that think of the sarcomere as yourself, and the myosin head as your hands, and you're pulling the the rope towards yourself. So the rope is the thin filament. So you're pulling the thin filament towards yourself, and you is the hands. You're pulling the thick filament is pulling the rope towards itself. It's pulling the what do you call it? The thin filament towards itself, and so it's pulled towards you, and therefore it's contracted, right? It's contracting. I mean, more the distance. I mean, the more you pull it, the more contract it gets from one end, right? From this end, it's gonna get more stretched, and you're gonna pull it towards yourself, so it's gonna keep on getting. There's gonna be a lot of tension here. So, when this happens, um, the ATP it attaches to the myosin head, therefore causing the myosin head to deattach from the actin filament. And when it deattaches, I mean, how does it deattach? When the ATP hydrolyzes, and when the ATP hydrolyzes, it releases energy. This release of energy causes the myosin head to leave its position, leave this position, and go back to its original position. When it goes back to its original position, the cycle continues until the process is done. So this is how basically the sliding filament theory occurs. Hope you guys understood it, and this was today's nervous system part two. And thank you so much. And I've been reading all of your comments, guys, and I mean, I swear I'm really happy, and this progress is really motivating, and I'm gonna continue these videos, and I hope you guys are having a good day. Until the next video, thank you so much. Bye bye. Take care.